when we talk about making mRNA or making a protein translation, what we're talking about is taking that mRNA that was transcribed in the nucleus, right? Because DNA does not leave the nucleus, all right? There is DNA in the mitochondria as well, or chloroplast, but in general, in a eukaryote, the genome, the genomic DNA is in the nucleus. Is that clear?
Yes.
So the mRNA leaves the nucleus and goes into the cytoplasm. It has a sequence. There were some changes. Transcription wasn't that simple. Hold on, let me say this again. I'm just going to say it all on record so that I don't want to have anybody, any miscommunication. The people in the back that are not paying attention, I'm going to ask you to move. I'm going to ask you to move on your own. I need you to move up front somewhere, and I need you not to waste class time anymore. Please and thank you. So what we have here is a long sequence of RNA that was copied, right? When I say copied, I mean what do I mean by that? Is this identical to the DNA? What is this? It matches, right? It's complementary, right? And there's some changes because wherever there should be a T, there's a U, right? So this is complementary. So that's the first thing that's different from the DNA. So that region of the DNA, that gene that you transcribe, this is very similar to it, but not identical, even when it was first made, right? Then after you transcribe it, but before it becomes a mature mRNA, the very first thing that happens is, or one of the first things that happens before it even gets out of the nucleus, is you take out what? Let's see who's been reading. You've been doing this lab for a while. What do you take out from the mRNA? You didn't do it in the lab. You didn't do it in the lab because it said, we're not going to do this in this lab, but don't forget, something comes out of the introns. Good. The introns. Yeah, the non-coding regions. If you watch the video, I explain it. I already sent it to you. I even put music to it. And it also explains it to you in the reading in the lab. So the lab does say it. It's okay, we're going to move on. So the introns are taken out. But I agree with you, it seems like it's backwards. It's not, but it does seem like it's backwards. So, introns, son of a gun. Ow, that hurt. Yeah, it's one of those days. All right, so you take out the introns, that's the first thing you're going to do here. And then after the introns are all out, you still have to put a GTP cap, right? And a poly A tail. And so after you've made all those changes, that makes this different from the gene, right? So already the gene is not identical. Already there's a real big difference between... All right, so the sequence here is already different from the gene, right? Instead of T's, there's U's. This is complementary from the gene. There's a GTP cap, there's a poly A tail, and the introns were taken out. So we call those modifications, right? So it is the gene, a version of the gene in an mRNA form that's been modified. We've changed it. All right. So it leaves the nucleus, it goes into cytoplasm. These are important points. I hope people aren't doing their homework now, because this is what I'm getting. This is what I'm getting a sense that you're doing your homework right now and getting help with your homework right now instead of listening to what's going to be on the test. What should have happened is you did your homework over the last week and a half to do a chapter. Should have done your homework, answered all the questions. That should be on the table. There shouldn't be people in the middle of class trying to do their homework last minute. That's not going to do you any good. That's going to do you zero good. You're not going to pass the class anyways, because you're not doing your work when you should be doing your work. I'm just letting you know. Why do I say you're not going to pass the class? Because you're going to fail the test. Why are you going to fail the test? Because I'm explaining stuff to you right now, and you're not listening. You cannot pass a test that of course you don't understand. When you come to me and say, I don't understand, of course. How do you understand? You're not paying attention. So, all right. So you have an mRNA strand. You took out the introns, you changed it. It's out in the cytoplasm. It meets up with a ribosome. There's 3 spots on the ribosome. The book and the lab give you the letters, right, that are corresponding to those parts. I don't really care. I'm not going to give you the letters. I just need you to know there's 3 spots. Why do I mention that? Because that's how the ribosome reads it, in 3s. Everything's in 3s. The code, the triplet code, the 3 letters sequence on the mRNA that the ribosome reads is called the codon. Codon, C-O-D-O-N, right? Everyone here should know that by now. 1, 2, 3, 4, 5, 6, 7, etc. So each 3 is a codon. When this matches up with the ribosome doesn't start. There's no bonding between tRNAs and mRNA until you get to AUG. Is that clear?
Yes.
That's why we call this the start codon. AUG is the start. Look at the codon sheet, the chart on the back of your lab that you should know. They're all there. They're all there. You're always, in my class, you're always going to have the chart. AUG is the start, and what the amino acid that's carried by the tRNA, listen to the words I'm using carefully. The amino acid that is carried by the tRNA that matches AUG is called methionine. So AUG codes for what? Now there's 2 ways you can look at this. You can look at this as the AUG portion, or you can look at it as a complementary anticodon. So you can look at the codon or the anticodon. Almost every chart I've seen, and I've heard of the anticodon charts. I don't particularly think they're important for us. But almost every chart I've ever seen, used on any test that I've seen, you're only going to use the mRNA codon. If it says codon, you're looking on the mRNA. You're not looking at the DNA. You can't use the DNA to figure this out. Is that clear? Because of all the changes we just talked about, right? You have to look at the mRNA. You have to turn it into mRNA first. In a problem on a test, if they tell you that you have to change it to mRNA, and they don't, and they give you spots like they do in the lab, and they tell you what does the mRNA look like, right? Then you have to assume that they're not taking out the introns. Does that make sense? Because even though we know that process happens, often we don't bother with discussing that on a test because they're not testing whether you understand that introns are taken out. They're testing, do you understand that if there's an A on the DNA, there's a U in the mRNA. They want to know, do you understand that the code on the mRNA is equal to an amino acid? That's what they're testing. That's what you will be tested on next week. That's what's going to be tested on the end of the course exam. They want to know that you understand that when we're talking about translation, the codon, the triplet code on the mRNA, is equal to an amino acid that leads to an amino acid sequence that makes a protein that does something. It could turn out to be the protein that makes your bones. It could turn out to be the protein that lets you see. It could be the protein that changes your skin color. It could be whatever protein that the protein does. Proteins have a function. Is that clear? Yes. It could be structural. It could be enzymatic. It could be it could be a pigment. It could be a lot of things. Is that clear to everybody?
Yes.
No. What is happening right now? Why can't I just say no and someone say okay, no? Thank you, Mr. Vanessa. I'll ask. Okay. So AUG is the start codon, right? And whenever this ribosome complex sees an AUG, there's a tRNA. The tRNA lands, right? It lands, it matches, you see they match, the hydrogen bond. These are nucleotides, just like with two DNA strands. These are two RNA strands. This is tRNA, right? tRNA. You see in your lab that they drew the tRNA, right? If you look carefully, you'll see a tRNA. It's actually the shape of a T. Then they ask you, what are the letters? Why do they ask you what the letters are? Because you have to match them here, don't you?
Yeah.
Right.
Oh, so when we put.
So when you're putting those letters here, how do you know what amino acid to put here? Yeah, you look at the letters on the mRNA, AUG. And you go to the chart. Don't look at UAC, because in fact, in the lab, they don't even write UAC. You have to write UAC.
Right.
But don't you go looking for UAC on a codon sheet, because the codon is AUG. If you look up UAC, that's wrong. In fact, I pointed out that they made that mistake on the lab, didn't they?
Yes.
They made that mistake in the lab. They actually used the anticodon to look up the amino acid. And another part of the lab, they used the DNA to look up the amino acid. That's a mistake. Those are major mistakes. It's a beautiful lab, but they just made a few mistakes, so you have to correct. I think it's a great learning opportunity, right? Because when somebody else makes a mistake, that's an opportunity for you to learn. That's why tutoring people helps so much, because then you have to explain it. You have to find a way to understand it to be able to explain it to somebody. So the anticodon matches the codon. This specific tRNA carries methionine. This one does no other. This carries nothing but methionine. Is that clear? And this one only carries what? THR. And this threonine. And this one only carries arginine. Right? So these are very specific. So AUG, there's going to be a trick to this for you to understand mutations, so pay attention. And obviously we're going to have to spend the whole day today just doing this, to explaining, and then we'll finish the lab tomorrow, right? So the methionine, when they're in the ribosome, the methionine bonds, makes a peptide bond, you see it right there, to threonine, and then the next one, and then the next one, and eventually you end up with a long protein. That protein is going to fold, right? It's going to fold into some kind of shape. That shape is going to define an enzyme, or it's going to define a pigment, or it's going to define a structural protein. It's going to define something. It's going to have qualities that allow it to do its function. Does that make sense? So if you change this protein, you've changed the function. It's possible you change the function if you change the protein. Maybe you change the amino acid, it really didn't change the function much at all. It could make the function better, or it could make it worse. A mutation is not bad, right? It could lead to cancer, but it could lead to you being stronger. A mutation is just a random change in the DNA sequence. That's all it is. The results of that depend. So let's look at one of the... So transcription is when you're taking your DNA, and you're doing this in the lab. You have your DNA, A, T, G, C, whatever it is. It's very long, right? And when you're talking about transcribing, what is it that you transcribe? What part of the DNA? The part of the DNA known as the gene. A gene is not the whole DNA molecule. A gene is a portion of the molecule. So this part right here this part right here is the gene. Okay? A gene is a part of the DNA molecule, a part of the chromosome. Remember, a chromosome is a DNA molecule that's been all wound up. But a part of the DNA molecule that has what? That does what? It undergoes what? Transcription. So if there's a mutation up here, if you change the A turned into a T up here, will that change anything? If there's a substitution outside the gene region, would there be a change in the protein? Huh? Okay, I'll try it one more time. Number one, a gene is the part of the DNA that is transcribed. So obviously, based on your lack of answers, you don't understand that transcribe means you're taking DNA and turning it into mRNA. And eventually, it's going to do what? Go through translation. And it's going to turn into a protein. The protein is the thing you see. The protein is what makes you big, small. Hold on a second. You know what? I'm going to leave it recording. I asked you to take the jacket off. Please take the purple pink jacket off. Please take it off. So DNA, the mRNA, the protein. That's the part, the gene is the part of the DNA that gets turned into a protein. That's what makes you big or small, or changes your hair color or texture, changes your face. All the characteristics, those things that we call characters that make you who you are, is because of the protein. But what made the protein? The mRNA. What made the mRNA? The DNA. That's why we say the DNA controls the cell, because everything that the cell does, and by extension, everything that this body does or is, at least in part, came from what? The DNA. Is that clear to everybody? I hope it's clear, because when I ask a question in class, I get silence, right? So I need to know, do you understand this or not? I don't want to give you a test and then you not know the answer. So a gene is a part of this DNA that makes a protein. So if there's a mutation, what is a mutation? What's the definition of mutation in general? Kind of general idea of a mutation. A change in the sequence. So as a base is changing. So instead of A here, what do you get? A T. So the A turns into a T. If it turns into a T here, then that changes the A, that changes this one over here to an A, right? So you get, for every time you change one, you have to change the other, because they only match, A only matches with T. Am I clear on that? Would a change outside of the gene, would a mutation outside of the promoter and the gene and all that, would that change the protein? So if I change this A to a T here, look at where I'm pointing. I'm pointing here. If I change this A to a T on this end, the gene is here. This is not a gene. This is not the promoter, right? It has nothing to do with making this protein.
Right.
If I change it from an A to a T, will that change the protein?
Yes.
Why? How? How is a change here going to change the protein? It's not going to change. But I'm wondering if you're saying it's not going to change because I questioned you, or do you actually understand?
Because it's not related to, it's not on the gene.
So if it's not in this region of the DNA, then it's not making a protein. So if you change it here, change it from one to the other, you have a substitution, then it's not going to change the protein. Does that make sense? No, as long as it's not the gene or any part of the gene function, the functioning parts of the gene. Can we take all our things off of the table? Because people insist on using their phones in class, and I need to know who's paying attention and who's not. Because when I have conversations with some of your parents, I don't want the parent to tell me, well, how do you know my kid is this, or whatever the argument's going to be. I just want book bags off the table. I just want your notebook open. I want your attention. I don't want you putting lip gloss on or texting or whatever else is going on. So what we have here is this idea that when you have a gene, it's going to code for a protein. A change in this region that we call a gene is a mutation that may cause a change in the protein. It may not. In fact, most mutations don't. And we're going to see today why. So gene is a part of the DNA that codes for a protein. Are we clear? Transcription is the making of the mRNA. Translation is the making of the protein. That's what you have. Right? So let's go ahead, and we're on this sheet where it says gene chromosome mutation worksheet. Please have your lab ready for submission to turn into me the physical lab with all the answers on the day of your final. Is that clear? On the day of your final. You're going to come in, you're going to put, there'll be the, I don't know where people learned that you can just scream out questions and answers, but when you come in, just place your lab on the front table, sit down, and you get your final, right? That's all that's going to happen. With a lot that and these mutation worksheets. Obviously, this one's going to be practice. It's the other one that you're going to submit for grade. Why do I say this was going to be practice? Because we're doing it together. I don't want to see, I don't need to see this. You don't want to do this, then you want to complain you don't understand. That's okay. But I'm not going to grade it because I'm giving you the answers. There's a lot of mistakes here, which I look at this as an opportunity for us to learn. So it says the substitution, gene mutations, as we discussed yesterday, gene mutations are affect a single gene, if they affect it at all, right? Because we know a mutation is just a change in the DNA sequence. Most mutations don't have an impact on the gene, and those that do change the gene don't necessarily change the protein. Does that make sense? So that's the idea that you have to, that's why it's important that you understand the process so that you can understand, well, what if this happens? Well, if this happens, then that's going to happen. And if this other thing happens, then it's not going to happen. Does that make sense? But in order to understand, in order to be able to answer the question, you have to understand how it all works together. You can't memorize what DNA is and not understand how it's replicated. You cannot memorize what a protein and what RNA is without understanding how you get things to go from a gene to a protein. If you don't understand how things go from a gene to a protein, then you're not going to be able to get the answers right on the final or in the end of course exam. And in general, you did not learn what you were supposed to learn. That is the objective. How do you get to go from DNA to a protein? So a gene mutation is a change, and one of the types of changes that could happen is something called a substitution. And in a substitution, what happens is you take a letter in the DNA and you change it to another letter from A to G. Now they made a mistake here, and the mistake they made is they changed the DNA, and then they read that three-letter DNA code. They read it as an amino acid. That's not right. Do you agree?
Yeah.
Can we do that now? Can we change this to RNA and then look at the RNA chart, the codon chart, and let's look at the change? I know most of us know because we started this yesterday, but some people were absent. So let's go through it. Let's practice doing it now together. All right? So when we're looking at G, what's the C in this CAG? What's it gonna make? G. And that's right. And what's the A gonna make on the RNA? U. The match. I say make, I mean what's gonna match, right? And what's the G gonna match with? C. So what you're gonna get in the mRNA, if this is the mRNA, what you're gonna get is CUC. Is that correct? Do you agree?
GUC.
I'm sorry, GUC, my bad. GUC, do you agree?
Yes.
All right, so if you're going to get GUC, what amino acid would you expect? You would expect valine, val, right? VAL. How do you know that? Does anybody in here have problems identifying where you go to look for that? How do you know that? I mean, from the chart. So you go to the chart. You're always going to have the chart. You don't have to memorize it. I'm saying that. I'm literally just said it. The chart is always going to, you're always going to have it. You're not expected to memorize all the codons and all their associated amino acids. That's ridiculous. Right? Not even in college are you expected to memorize it. Although you memorized a lot of them once you work with it for a long time, but you don't have to worry about it. GUC is valine. GUC on the mRNA. Notice I said on the mRNA. Their mistake when they did this worksheet, they put GIN. Why did they put GIN?
Because of the CAG.
Because of CAG. They read CAG and they put GIN. Look at CAG on the chart. What is it? Gin, yeah. That was a mistake because how do I know that's on the DNA? How do I know this is DNA? Because it has a T. As soon as you see a T, it's DNA. You know that. So that was their mistake. So that was valine. But the change, now we changed the A to a G. How did that change the RNA? What's the new mRNA? What's the new mRNA? What is it? Go ahead and do that. If you don't have it, do it. What's C?
G.
Is G. What's G?
C.
C. What does G match with?
C.
C. So what's GCC? Alanine. ALA, right? Alanine. So it's actually a change from valine to alanine. Do you see that? It went from valine to alanine. Do you get that? Does everybody understand the change there? You have to make sure you change it to an mRNA first. When you change one, now hear this clearly and write it down, and it's written here for you correctly. If you change one base from an A to a G in the DNA, that's a mutation because you changed the DNA. You have to change the DNA in order for it to be a mutation. If you change one base change is called the point mutation. It's a change in one point of the DNA. So it's called a point mutation. A single base change. You see that? All right. This is also known as a missense. Missense. M-I-S, sense. It's right here, right? So this missense mutation, what does it mean by missense? It means that we change the meaning. Mis means like misunderstanding. Sense means understanding. Like misunderstandings, you missense is a misunderstanding. So you went from, it's supposed to be valine, but now it's alanine, so we say it's a missense. All right? Anytime you change the amino acid sequence, it's called the missense. Anytime you change the DNA that leads to a change in the amino acids, it's called the missense. It's not a very common thing to happen. These mutations don't happen as often as you might think. So let's see. I wonder if they did these right. I didn't check. Who looks at T? Look up. There is no TTC, EGT. There's no TGT, right? Mm-hmm. So it would be A,
C.
Hold on, let me change the color because it's hard to see. It's going to be ACA. What's ACA? THR. So I don't know how they're getting cysteine. How'd they get that? There's no TGT.
Right. Hold on.
Maybe they're using a different chart. Maybe they're using some kind of DNA chart.
The only way you can get that is UGU or UGC.
I think they changed it to
I don't know. I don't know what they're doing, but maybe there's some kind of, I don't know how they're doing this because it doesn't make any sense to me. But yeah, whatever they're doing here they're doing it incorrectly. So as far as I can tell. So ACA is what? THR. Right? Oh, that's not how you do it. THR, right? Everybody agree?
Yes.
So over here we changed ACA to what? ACA. G, which is what? THR. So it turns out that it's still a silent mutation, isn't it?
Yes.
Why do we call it a silent mutation?
Because
That's exactly right. The amino acids stay the same. Excellent. Because in their mistaken way of doing it, their amino acids stay the same. But you see here, the amino acids stayed the same. So it's still a silent mutation. So weird that I don't understand. Maybe I'm misreading something. I don't know. So, but there's another type of mutation called insertions or deletions, right? What do you think an insertion is?
and something's coming in.
Adding something in, right? I almost want to say a duh, right? It's kind of straightforward.
Yeah.
So if I change this from a C to a G, but I don't put a new one in, that is called the point mutation, right? Or substitution. If I insert something new, now this is the trick. The trick is that this is all read 3 at a time. It's not one after the other. So by reading, so if it was one after the other, if this was alanine and this one turned out to be glycine, and then this one was glutamine or whatever, right? You would just get an extra amino acid in the middle. But that's not what's happening. You're reading them every 3 letters. So as soon as you insert something new, you changed everything after that, right?
Yeah.
You changed that codon and everything after that. Now this is the DNA, but when you make the mRNA, you're going to change the codon, right? So when you look here, you say normally it would be a bunch of histines in a row, right? Even though the letter, it's a repeat, you agree that's a repeat, right? CAT, CAT, CAT. You get a bunch of histines in a row in this particular section of the protein. Here in the new mutation, when you insert a new one, now all of a sudden you get 3 histines, and then everything after that changes. So that's called the frame shift. And the reason they call it a frame shift is this. Think of reading this with a frame. Let's put a frame around the 3. And you take this frame and you move it every time you read it, you're moving it. So you have to think of reading it as if you had a reading frame. In other words, you had like a little box. You ever see people use like their finger to read across the page?
Yeah.
Well, imagine using a little box and just moving the box around the page and reading it only what's in the box. It helps people focus. It's a method of helping people focus that can't, look at a whole page of words and freak out. Well, if you just take a box and put it around it, then you're just reading one word at a time, and you can read it clearly, and it simplifies things for some people. Do you agree? Do you see how that would work? So here, if we use this as a reading frame, so we put a frame around it, and we just move it 3 at a time, it's easier for us to read it, right? If we were going to read a big, long, and by the way, we used to do that in the lab, we'd have to read a bunch of letters, we would use a frame to help us make sure we don't confuse them, we just do them 3 at a time. Does that make sense? So that's our reading frame. So now, if we insert one, what did we just do to that reading frame? We shifted it, right? We shifted it by 1 because we added a new one. So we used to be 3 and then 3. Now we put a new one in. Oops, that one's not that 3 anymore. It's the other one, right? So let me highlight this by going in and doing the box here. So that's 3, right? Let's draw the other one, and there's another 3 here. Do you agree? And then there's another 3 here. And then there's another 3 here. Each of these is CAT, so each of them is histine. So now we do the same thing down here. Let's do it in blue, or not green. It probably shows up better. I don't know. So there's 3, right? And there's another 3. That's easy. And there's 3. So still all histine. But now what happened, now instead of reading CAT, you'll notice here that this is still CAT, right? That would be histine, wouldn't it? If it's histine. I don't even know if it's histine because it's not histine. Okay, so whatever. So if you read it, it's now ACA. And then the next one's what? TCA. And the next one. So now you shifted the reading frame. So now all the 3, instead of being a repeated unit of CAT, just by putting one new nucleotide in there, DNA nucleotide, just by adding an extra A, you shifted everything over. So all your CATs aren't CATs anymore, they're TACs.
TCAs.
Or TCAs. Huh? It didn't go anywhere, it just kind of moved, everything moved over. So it's like we're all in a line, we're all the same, and then you have 20 boys in a row, and every 4 boys equals $1, right? Every 4 boys in a row equal $1, but then you put a girl in there, you just mess things up, didn't you?
Right.
So just because everything shifts over, I don't know, I just made that up, it didn't seem to make any sense. All right, that's good. All right, so that's one way of shifting the frame. There's another way of shifting the frame, and that's a deletion. So this is an insertion. A deletion means you take one out. Now see a CAT, CAT, CAT. Now you take out the A, now it's what? CTC. And then ATC, ATC. So you get the same kind of impact, don't you? You get the same kind of, you shift the frame over, the reading frame, by this time taking one out. Because it's all read 3 at a time, right? So if you mess up any one of those, where you're taking one out or putting one in, then you can mess up everything. If you just substituted one, then you just change one amino acid. You see that? But if you shift things, then if you take one out or put one in, now you shifted everything. Does that make sense to everybody?
Wait, say that again?
If you insert or delete, then you're shifting everything after that. If you're just substituting, then it's not as drastic a change. Does that make sense?
Yeah.
All right. A substitution would change one. So now let's look at these questions here. It says there are several types of gene mutations. List 2. What do they have in common? How are they different? So you should be able to tell me, right? So do these. Make sure you know them because you're going to get questions next week. You know how many exactly, right? You're going to get on mutations. So know your point mutation, know your deletion, know your insertion, know the difference between a point mutation and a frame shift, right? There's one an insertion, a point mutation is just a single substitution, for instance, just a substitution, right? Does that make sense? You substitute one. And a frame shift can either be a deletion or an insertion. It's just that simple, right? Okay, so, yeah, we can talk later, but yeah, a frame shift can either be an insertion or a deletion, right? So you should be able to write that out. I shouldn't have to write that out for you. So list 2 of those and explain how they have in common. The geneticist, if you don't do this, I'm not going to grade it, right? I explained it just a second ago. It's recorded, I'll post it. So I'm not concerned about, if you don't do it though, if you don't actually try to do it on your own to make sure you can do it, then if you don't know if you know it or not. Does that make sense? A geneticist found that a particular DNA molecule had no effect on the protein coded by a gene. What kind of mutation was this? A geneticist found a particular DNA mutation had no effect on the protein coded for by the gene. Yes, it was a silent mutation. Absolutely right. Hold on a second. Let me insert a chart. It's called the universal code, right?
How do you read that?
Well, I'll explain it again, but just give me a second. So, how do you read the chart? It's a good question. You start on the left.
First base.
Start on the left. This is the first base. So if you have ACU, A is first, right? So that you start here, A. It's just like a game of Battleship. Literally, it's just like Battleship. I don't know if you play Battleship, but if you haven't played Battleship, maybe you should. It's fun.
But the other letters aren't
A,
over?
and let's say C, right? Next one's C. So A, C, so it's this row because of the first one, A. It's this column because of this C, right? This is the second one. The third one's one of these, ACU. There it is, ACU. Read. Read it, right? It says A, then C, so it has to be in this box somewhere. There it is. Now, if you had on some charts, so the ones that you have in your homework, for instance, they have all the letters here, all the letters here, and all the letters here. But oh my goodness, guys, once you get it into one box, there's only 4 possibilities. All right, so ACU would be 3 and 8. So this is kind of the short version of it in this case. Remember, this is all RNA. These are all codons. You have to read the codons. You can't go and try. Now there's something else. Well, I guess we'll wait till the next one. But let's take a look at this. It turns out, how many different ways can you get leucine? Leucine, L-E-U.
4.
No, 6.
Where?
6 there, 2 here, and 2 and 4 here.
Oh.
6 ways to get leucine. So it turns out, if you had a change, if you changed the DNA, even if you did a point mutation and it was in the gene, right? If you did a point mutation and it was in the gene, it still doesn't guarantee you're going to get a change in the DNA, in the protein, because of what we call, hear me clearly, because you may see the word on the test, maybe not, but you might see it on the test, redundancy. Redundancy. What do you think that word means, redundancy? That's right. That's not hard, I guess. Redundancy. I'm writing with my finger, my pencil. So redundancy, the ability, the redundancy is the tendency that there are repeats. So you can make a change. Let's take a look just as an example, then we'll look at actual examples. If you had something with CUU, you could literally change the middle one to, or not the middle one, you can change the last one to whichever one you wanted, and you'd still have leucine. If you had CU, right, you know it's leucine. There's no other choice, is there? All CUs are leucine.
Yep.
Whether it's CUU, CUC, CUA, or CUG, it's all leucine. So you could mutate that last, you could point mutate that last DNA, right, in the gene. This is the RNA, but in the gene you can mutate that last nucleotide to any nucleotide you want, and it's still going to be leucine. Does that make sense? So there's a lot of redundancy in the DNA, in the code. So that's another layer of protection for your DNA. So let's go ahead and look at let's look at the this question. Wow, I would expect to see something like this on the test. I'm telling you. I think out of 30 questions, I've already told you, I've given you the test blueprint, and this week, if you've been paying attention, I literally have given you like half the test. I've given you what's going to be, and this is an example. It's not going to be exactly the same question, but this is an example of one of the questions that's coming on a test. So here we have it. 2 of these code for the same protein. Which one? So let's go through it and do it. Are you ready? You have your charts. You guys are going to have to tell me. It's in your lab. It's on the back of your packet. It's also in your lab. Hold that up so people see it. If you don't know that it's there, I'm concerned that you're not actually doing your work. You can sharpen it back there if you want, but do it quick because we're about to go through this. All right, so let's do this. AGU. Now, is this the mRNA? This says, what is this? This is what? mRNA. So now we can go directly from this, right? But if it was DNA, you have to change it to mRNA first, or it doesn't work, right? Don't make that mistake. AGU is what?
Sir. S-E-R.
S-E-R, serine. Let's see. Let me see if my pencil's done recharging, because this can be. S-E-R, serine. Okay? So what's UUA? Now notice I'm doing 3 at a time. I want to point that out. You have to do 3 at a time. That's another mistake some kids make. 3 at a time. Now, also one other thing. On some problems like this one, they're not going to bother with putting in AUG. But be careful. If they give you a big mRNA and they say, what's the protein that this is going to make? You have to start at the first AUG, from the left to the right, the first AUG you see in a row, AUG. That's your beginning. So be careful, all right? In a problem like this, that's not an issue, right? Because there are no AUGs. They assume that this is in the middle somewhere of the RNA. You have to assume that. All right, so what's UUA?
L-E-U.
LEU. What's Should we do all three of them at the same time? What's UUC?
UUC.
I'm sorry, UCG. Sorry, my bad.
Sir.
SER. So that's still, look at that. These look completely different, don't they? How is this the same as this? Because of redundancy. Right? Look at your chart. You see that more than one mRNA codon.
Where is sir? Oh, it's here.
You see that?
I thought they would be like.
Yeah, see, is that crazy? So you get, this is a mutation. This has been mutated. If AU gene was the original, you have a mutation. But do you see a difference in amino acids?
Yeah.
No. They're both serine. So what do we call, what kind of mutation is that? Silent. Silent mutation. They have the same amino acid because of redundancy. Redundancy. So there's more than one way to get serine. So you can change the code. You can change the DNA, which changes the mRNA. But because there's more than one way to get serine, you're still keeping the same amino acid. All right, so now let's go ahead and what's the next one? AGC.
Sir.
SER. Another crazy change. And so there's three different versions of the gene give you the same amino acid. Do you see why it's so hard to mutate the DNA?
Yeah.
Number one, the DNA polymerase self-corrects. It corrects mistakes. That's number one. That's the first layer of protection. Well, actually, probably the first layer of protection is all the DNA is kept in a nucleus. It's protected over by an entire cell, right? Secondly, you have a DNA polymerase, an enzyme that corrects mistakes. So if you change an A to a T, normally, almost all the time, your DNA polymerase will change it back. Thirdly, only the gene, only the section that's the actual gene, only a change in that, usually, only a change in that will change the protein, right? On top of all that, if you have just a substitution, and it does change the amino acid, it's only one amino acid that's changed. Then another layer of protection is that you have this redundancy. So if you have 5 or 6 different ways of getting the same amino acid, that means you can mutate that area, that specific codon, those specific nucleotides, 6 different ways, and still get the same amino acid. That's a lot of protection. That explains why all our DNA has been very similar for so long. Otherwise, there would be a lot of changes, right? Like I said, like the example we used earlier in the week, if I gave you a dictionary to copy by hand, and assuming you could move through time quickly and do it quickly, you would not be able to do it without making mistakes. So it's important that the mistakes don't impact the meaning, and it's important that we correct mistakes. Does that make sense? So, all right. What's the CUC? What's CUA?
LEU.
LEU. So still, and what's CUC?
LEU.
Look at that. Isn't that crazy? Yeah, you have a question? On what? Redundancy? It just means repeats. More than one way to get the same thing. Redundancy just means more than one way to get the same thing. Listen, NASA, when let's say you were an astronaut. Let's say tomorrow we're going to put you on the moon. We're going to put you on the moon. Well, we're going to shoot the rocket. You're going to get in a rocket, right? And we're going to blast some number of tons of explosive underneath your butt, and we're going to shoot you up into the sky, past the sky, into space, past space, to the moon. You're going to orbit the moon. You're going to get in a little craft that's part of your spaceship. You're going to blast off of that and go onto the moon. You're going to jump around the moon, collecting rocks or whatever, get back in that thing, lift off, link up to your little spaceship, turn that ship around, come back to the Earth. You're going to come skimming off the atmosphere of the Earth. You're going to come flying down. It's going to be so hot that the outside of your spaceship is going to turn red. If it wasn't made of ceramic, it would melt. and then a parachute's gonna open once it slows down enough, and you're gonna float down to the ocean, and you're gonna land in the ocean. There's gonna be a little explosive gonna bust out the door, and hopefully the Navy's there to pick you up, because otherwise you're gonna drown and die. So they'll pick you up from the ocean with all your rocks and take you back to NASA, where they're gonna give you a hero's parade, and you're gonna be an astronaut. Oh my God, well, you're so brave, blah, blah, blah. Let me ask you something. What do you think we do? What do you think NASA does? Because it's gonna take many days of you in space, right? You need oxygen, don't you? Do you agree? You need oxygen to breathe.
Yeah.
So we have oxygen on the spaceship. You need 1 tank of oxygen to go there and back. Why does NASA put 3 tanks of oxygen on the spaceship? If you only need 1,
Extra.
in case something goes wrong, they do what? They are redundant. NASA uses triple redundancy. They have 3 times the amount of whatever, because just in case. Does that make sense?
Yep.
What part of that does not make sense?
What are you talking about? They have 3 times a month.
All right, I'm gonna try one more time, then I'm gonna move on, okay? You can see me after
You don't
school.
gotta say the whole entire thing.
No, I won't. I'm gonna try to make it short. So, let's say I'm gonna put you, literally, think about it. I'm gonna put you in a spaceship and send you millions of miles away in space, no air. If that's too hard for you to imagine I'm gonna put you in a submarine, a mile underneath the ocean. Why would I give you 3 times the amount of oxygen that you need to breathe?
Oh, I didn't hear the oxygen part. I just heard 3 times what. That's why I said.
Oh, okay. 3 times. Most things in this kind of system. Look, tankers, ships that move oil from one place to the other. You only need 1 hull. 1 hull, right? The hull is the outside of the ship. You only need 1 hull. It holds the ship up. It works fine. Why do they double hull it? Why do they put 2 layers between the ocean and the oil? In case something goes wrong. So that's what redundancy is. Redundancy is having more. Why do things happen? Why are there 6 different ways to get leucine? Why in this code are there 6 different ways to get leucine? Because it helps protect the DNA. I mean, that explains why this has lasted this long, right? That there's 6 different ways to get the same amino acid, because that protects against changes that are inevitable, right? There are going to be mistakes in the DNA. That's not correctable. That's not something we can stop, even though we try. I'm not having 100% focus on this, so I'm going to assume either everybody understands or you don't care, so I'm moving on. All right, so GCA, what is that one? ALA. What's GCG? ALA. And the last one's ALA. So all these are ALA. So you guys went ahead and did it because you got it. Okay, good. These are all THRs. Okay. So everybody seems to have it. I hope you're the same way on the test. And so these are all SER, SER. You just heard him. You just heard it. Thanks, man. You just dropped me in the middle of the hot soup over here. All right, so go ahead. ARG. SER. ARG. So this is the one that's different, isn't it?
Yes.
So we've answered the question. But let's finish it off. What's the last one?
SER.
All the way to SER. My God, guys, could there be a clearer example of the fact that you can change the DNA many different times? This is RNA, right? But to get these different RNAs, you had to change the DNA. You can change the DNA many times and still get the same amino acid sequence?
Yes.
So it's very hard to get a mutation to actually change something. When a mutation changes something, changes the protein, it can be good, it can be bad. And number 7, make sure you answer these questions. They're very good questions. But let's look at an example of sickle cell anemia. Let's look at sickle cell. Again, please ignore the book reference. It's a different book. So, but,
Yeah, we don't have the book reference.
I know. I'm just don't want any people coming to me and saying, I don't know where this book is. What pages? We don't have those pages. It's not a different topic. No, it's not relevant. Below is a DNA base sequence of the normal protein for normal hemoglobin and the base sequence for abnormal. They call it abnormal. I don't like that. Abnormal, listen, it has a bad rap. The word has a bad rap. But I don't like to use the word abnormal personally. But I gotta say, I'm gonna take it out of the reading. Abnormal just means, normal just means average, 70%, right? In the middle of a bell curve. That's normal. And abnormal originally started with, you're not in the normal range. You're either really intelligent or really not so, or you're really pretty or you're not so pretty. And so you're not part of the average, right? Or you're tall or really short, you're not in the average. Does that make sense? But abnormal is turned into really a bad word, right? So let's not use that word. Like we could use oh, what the heck? I don't know what word you use, but let's say we have, what we like to say is, we like to say wild type. Wild type means that this is what you find in nature, right? And versus the non-wild type, right? All right, so, but rather than you even saying that, let's go ahead and just say, what do we want to call this instead of using normal gene and abnormal gene? Let's say non-sickle cell and sickle cell.
Yeah.
Non-sickle. I like that better, right? But the problem with me saying that, we say normal, right? When you look at the genetics of a population, the normal, about literally the normal, 3/4 of the people in various populations have the sickle cell gene. So why are we calling the normal non-sickle cell? Does that make sense? Where the majority of the genes in that particular population are going to be sickle cell. That's because it gives you protection against malaria. Malaria is a disease that kills people. It's a parasite. Having the sickle cell trait gives you resistance against that parasite. That's why most people in an area where there's a lot of malaria, they have the sickle cell trait. That's why African Americans and people from Africa tend to have a lot of these genes, because malaria is a real problem in a lot of places in Africa, but also in the Mediterranean and in South America. So to call non-sickle cell normal, it's not quite accurate. It's not quite accurate, right? So the other issue is that it's an advantage in places like having the sickle cell gene is an advantage in places that have malaria, but having this 2 copies of that gene is a problem, because without treatment, a lot of those kids end up suffering or having early death. As I said, my best friend in high school died from sickle cell anemia right after high school and college. So I can tell you from personal experience is that it can be bad as an outcome. He lived a good life, though. It was just too short, right? So having sickle cell, I see your hand, write it down because I want to get through this. Sickle cell trait is an advantage. Sickle cell disease is somewhat of a disadvantage, obviously. So 50% of the population have the trait, 1/4 of the population have the disease, or both copies. When I say disease, sickle cell disease is different from the sickle cell gene. Sickle cell disease is what happens what's the consequence of having blood cells that look like this instead of blood cells that look like this? It clogs up, they clog up, they stick to each other, they get stuck in capillaries, they cause a lot of pain, there's blood clots, they get strokes, they get all kinds of issues. People that have the disease have a lot of these cells versus this, right? Instead of just a little bit misshapen, they have this awful, it's an awful, painful stuff that happens to them because of the gene. But the gene itself is an advantage. It's when you have two copies of them that it becomes a problem. And today we have a lot of management, disease management. We still haven't found a cure yet. We're hoping that CRISPR and some of the gene sequencing technology that we've developed over the last 5-10 years, we're hoping that that's going to be the solution to the problem. We're hoping you're going to be able to go into a clinic, they're going to take some bone marrow out of your bone, inject you, they'll mess around with the genes and re-inject it into your bone marrow, and boom, you're cured. Because these cells are made in your bone marrow, right? They're inside your bone. I don't know if you knew this, but your bone is hollow. Your bones are hollow. Not really hollow, but they're hollowish. Because inside the middle, there's this mesh, this bone marrow, and in here's where your blood cells are made, right? So, yeah. That's some of it, yeah. There's a lot of issues. The bone, what happens is that in order for your bones to stay nice and dense, strong, you have to use them. This is a real rule of thumb, like dissolves like for instance, like high to low, right? Another rule of thumb in biology is if you don't use it, you lose it. So if you're not walking around on your bones, then your bones get thinner and thinner. Because you have to have mass on it. The bones, there's a reaction to having force on the bone. The bones get thicker and stronger. That's why women, as they get older, they're told you start to lose calcium, especially postmenopausal. So they tell you exercise. What does exercise do? And drink a lot more calcium. It strengthens your bones because when you're running or when you're pushing, and not just exercise like on a bicycle, because that's low impact. You want impact. You want force. Because you want the bones to say, oh, I got to get thicker. You want to stimulate that thickening of the bone. All right, so, and in space, the opposite happens, and so you lose bone density. We call it bone density. All right, so anyways, let's look at the gene. The non-sickle cell gene looks like this. Today, because of all the treatments for malaria and our good sewage, and especially if you live in Cleveland, this gene is an advantage, right? But if we were in Africa, especially in an area, or in South America, or in the Mediterranean, especially in areas where there's a lot of malaria, this gene would be an advantage. So which one's good? It depends. So it's not, there's no good, so when people tell you we're evolving to a better place, or we have this advantage or that advantage, that's not true. All that depends. So when we're talking about having an advantage, it's having an advantage for a specific condition. So if you have the sickle cell trait and it gives you resistance to malaria, that's an advantage when there's a lot of malaria around, but it's not an advantage in a modern society where you have better controls on the mosquito population, so you don't have this malaria problem, so the sickle cell trait isn't an advantage any longer. It doesn't hurt you, but 2 copies of the sickle cell gene ends up being a disadvantage. So instead of using the words abnormal or normal, it's probably better to use the words, is it an advantage or not an advantage? So now let's take a look at this and see what kind of change would cause all these issues, these cultural changes, these things that shifted the human condition. The human history has been impacted by sickle cell. And so what kind of humongous change has occurred to make this evolutionary step, to make this mutation happen, and therefore give an advantage to so many people and allow so many people to live in a place where a lot of people would die. So let's take a look at what this non-sickle cell gene looks like. You have this sequence here, and of course you should know, is this DNA or RNA? Well, I hope you said DNA, right? So how do you know? It's because of the T's. So you have the sickle cell gene, also the word gene. If you see the word gene, you know it's DNA. So here are these DNA sequences, non-sickle cell and sickle cell. And it says you're going to transcribe them and translate this gene. Now, of course, this is only a part of the gene. The gene is much longer. But rather than having to transcribe and translate a few hundred nucleotides or 1,000 nucleotides, you're only going to look at this small section. And the reason that you're going to look at this small section is that the difference between non-sickle cell and sickle cell gene, that difference is right here in this small section. So if we take this and turn it into mRNA, how would we do that? Well, you would need your trusty codon chart, right? And remember, this is a codon chart. This is a codon chart. So this is not going to be, you're not going to use the DNA sequence to read this the way they did in the handout. Again, I'm not sure why they would do such a thing. So the mRNA codon sequence is going to be, if it's a normal, you know it's going to be for every G, you're going to have a C. So you have C, 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 and then you're going to have G instead of T, or T codes for A. So A, and then G, A again, right? And then A. So this normal, again, I don't like that word normal, so I'm going to change that to non-sickle cell, NS. And I'm going to change normal to NS. You're going to see normal used a lot, but I just don't like that word for human conditions. All right, so sickle cell, then the other sickle cell, the sickle cell gene is going to be C, 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 G. Now instead of A, you're going to have U, and then A again, and then everything else is the same. So if you look at it, the only difference between someone who has the sickle cell trait, or if they have two copies of it, the gene, is that this A has turned into a U. So we have what kind of mutation here? Well, I hope you said it's a point mutation or substitution. So there's no frame shift. So we're only going to deal with how many amino acid changes in the DNA, in the protein. Well, I hope you said one, right? So because it's a point mutation, there's only one of them. You're only going to have one change in amino acids, and that's going to be this amino acid is going to change. So look, you see GAA, that's the mRNA, and the normal, or rather the non-sickle cell gene. So GAA is GA and then A, and that's going to give you GLU. And so you have the amino acid here is GLU. Now I'm not going to put in the other. I guess I'll go ahead and write the other amino acids. GCC is, here it is alanine, and GAA is GLU again, and AAA is lysine. And if you see that in the normal, you're going to have GAA, which is glutamine, but when you go to the sickle cell, you're going to still have alanine over here. You're still going to have GLU over here. You're still going to have lysine over here. That GLU is going to turn into GUA, which is valine. So that one substitution is going to change the amino acid from glutamine to valine, and that change causes sickle cell trait, resistance to malaria, or if you have two copies, sickle cell disease. And so it's amazing to me that one single nucleotide, that one little mistake, even though they're rare, remember why they're rare, remember the reasons why This is going to be important for the test. The reasons why it's rare that mutations have an impact on outcome, right? And the first, of course, is that the DNA polymerase self-corrects. The second important concept is that any mistakes usually happen in non-expressed regions of the DNA. What does that mean? Again, remember that only genes are expressed. And what do I mean by expressed? I mean, they get turned into proteins, right? Only genes are transcribed and translated. So most of the changes, or most of the mistakes, just because of sheer size and frequency, you're going to get most of those changes happening in areas that are not expressed or not translated, transcribed, translated. Then, of course, there's the idea that we have redundancy in the genetic code. So what does that mean? Again, just as a reminder, there's more than one way to get that the same amino acid. So even if you do have a mutation that occurs in the gene that's not corrected by the RNA polymerase, if you have that, if it happens in the third position, for instance, here in the CU series, you're going to end up with leucine. And even if you have a mutation mutations occur in the first two, you're still going to get leucine if you have UUA, UUG. So 6 different ways to get leucine, and that helps protect against these random changes that could be and could have a negative consequence. So we have self-correcting of the RNA of the DNA polymerase. Any mistakes usually in non-expressed regions. So that means that most mistakes are going to happen outside the gene, so it's not going to impact. And if it's a point mutation, and of course you have the redundancy. So, and lastly, if it's a point mutation, it's only going to change one amino acid. Now, sometimes changing one amino acid has a big impact, like you can see here in sickle cell. Sometimes changing amino acid has no impact. So the impact of the mutation all depends on where, how, what, and of course, is it corrected? So it's important to remember that it's hard for a mutation to actually cause a change in the phenotype. Remember, phenotype is the expressed gene, the collection of all the genes that have been expressed, the things you see, right? So in what kind of gene mutation is this particular one? We already said it, it's a point mutation. And as a point mutation, it's, we have it's also called the substitution. All right, if the base sequence, blah, 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 instead in A, right, in the problem above, would, let me see here. So the next question is if instead of this sequence you had this sequence, would it still be hemoglobin? Would it still work as hemoglobin? Well, what would you have to know to find out if it's still going to work as a hemoglobin? You'd have to know, do you have this same amino acid sequence? So let's take a look at what you'd get. A lot of you, or some of you, are going to try to take this and use this code to translate it. That would be a mistake, because you'd have to do what first? You'd have to transcribe it first. So to transcribe it, you get it to turn into mRNA, GGG turns into CCC, CTT turns into GAA, then GAA, and then AAG. And now we have our mRNA. Now we want to look at what amino acids these code for. Well, CCC is C, C, and C turns out to be proline. GAA would be glutamine. GAA again is glutamine. AAG is lysine. And is this a working hemoglobin gene? Well, look at what it used to be. It used to be alanine, glutamine, glutamine, lysine. Now it's proline, glutamine, glutamine, lysine. Are these going to be the same? Is it going to be a working hemoglobin? Well, the knowledge that you have, based on the knowledge you have, you're going to have to say no, because the amino acid has changed, right? You've changed the amino acid sequence, therefore you've changed the function. Is this going to be a functional hemoglobin? The only answer you could probably give with the knowledge you have is no. Now, you could say it could be. It depends. It could actually be a better working hemoglobin. Maybe it works better than the wild type hemoglobin. Maybe something happened here that made this this proline change made the hemoglobin better. But we don't know. So you have to say that the function has been impacted. Yes, that would be a better question. Was the function impacted? And then I would say yes, it was. Okay, the last thing that we need to talk about when it comes to mutations and chromosomal changes, we call it chromosomal mutations. Remember, chromosomes are wound up DNA, and they are wound up around proteins. They're structures, right? And we've drawn them on the board. We've looked at them, and people have asked repeatedly. So that means that's why we can't have, why different species can't mate with each other and produce a progeny, right? Well, remember, a chromosome, here's a single chromosome, right? This is what it would look like. It could be chromosome number 1, or it could be chromosome 2, whatever, and it depends on the species. Some species of organisms have hundreds of chromosomes. Some only have, humans have 23 pairs. So when we're looking at chromosomes, we have these little regions that code for protein, and what do we call those? We call those genes, right? So this region here would be a gene. What gene is that? It all depends on what chromosome it is, what's the sequence, what it does. So there's about 10,000 genes in the human genome. So it could be any one of those. So you have these regions, and they're spread out throughout. And so most of the DNA, most of the chromosome doesn't actually code for anything. And what's a key point here is that the genes located on the chromosome are located in the same place. For instance, this gene here, maybe it codes for hemoglobin. Maybe that gene is going to be found on this chromosome in the same place every time. So if something happens to the chromosome, if the chromosome breaks, and you all know about breaking, you drop your phones all the time and shatter the screen. If in the process of copying it, if in the process of moving it, if something happens chemically and it weakens some bond and this breaks, if the chromosome breaks and moves around, or if it's duplicated for some reason, or if it's flipped, or if it's moved to another chromosome, if this area of the chromosome is moved to another area of a chromosome, then you have a problem, because then some of the cells, after you divide, remember we talked about this idea of cell division. We're not going to get in too much depth right now, but remember we have these cells. They go, let me draw a little bigger. They go from one cell, and it was in a previous video, I believe, and we move it on to into two cells. If we have this one chromosome, and there's two copies, one from mom and one from dad, right? So mom gave you one, dad gave you one. We talked about that and the need to to split those up. If we took that, though, and we made copies of it so that we can make two cells, because we have to make copies of the DNA, right, to make two cells. So we go ahead and make these chromosomes, and they're linked together through these proteins, right, these structures. If for some reason this thing here, this area here gets, in this area here, if you get deleted, what if this portion on this chromosome gets deleted? This breaks off and it's gone. So this chromosome now is shorter, smaller. But the other chromosome doesn't, right? So we make copies of the gene, and what are you going to get as a result? Well, you're going to get one cell. Sorry, my nose is really. You have one cell that's going to have a normal copy of chromosome. This is dad's chromosome number 1. This is mom's chromosome number 1. One cell is going to get one copy of the chromosome that's great from mom, and from dad. The other one's going to be good too. So one cell is going to have both chromosomes that are normal. The other cell, though, is going to have this truncated chromosome from mom, and from dad, it's going to have a normal chromosome number 1. What happened to those genes that were there? What happened to these genes that were sitting in that region, right? Because these are all the same chromosomes, so they have the same gene in the same region. What happened to the gene, this copy of the gene is sitting right here that used to be there? It's no longer. Well, then now this particular cell, has only half that chromosome, only has half the genes that were on that region, because that region's missing, right? That area no longer, it's not just one gene, there's probably 5 or 6 or a couple hundred genes on there. So those genes, all those genes, you only have half of that DNA. So where you can only make half as much of the protein. And so that can be a real problem if that protein is a neurotransmitter, or if it's the protein, the collagen, the stuff that makes your skin. If you only have half the amount of protein available to the cell, and then therefore later to the tissues, then you have a real big issue. And remember, this division is going to keep going. So as these cells divide over, and it depends on when it happens. If it happens early on in the development of the creature, then what you're going to get is more likely than not some kind of disease, right? Some kind of issue, some kind of abnormal, I hate to use that word, but we'll go ahead and use abnormal condition that's going to end up causing problems for that organism. So when we're looking at chromosomal changes, and this is again why two species really have a hard time mating and producing a progeny that can then later produce more progeny, right? One of the big issues is that each of them have different numbers of chromosomes, and since they have different numbers of chromosomes, the genes are in different places. Even though an ape has similar, if not the same, genes for heart tissue, if their genes are on chromosome number 1, their chromosome number 1, and our genes for that heart are in chromosome number 20, then, and our children have, and we mate with in the laboratory, they mix our DNA together, it won't work because the number of genes won't be separated into the various cells in an accurate way. And so you're going to have too many of some genes and too few of other genes, and the organism usually miscarries. So the miscarriage is one of the first consequences, one of the earliest consequences of having a chromosomal abnormality or chromosomal mutation. So when we're talking about chromosomal mutations, we're talking about things like deleting portions of the chromosomes. So here you have this region here is there, over here it's not there, right? So all the genes that were here in this region are gone for that chromosome. Yes, there's another chromosome that has that region, but that could only mean half the DNA, half the protein in the end. You could duplicate it, which now you have more copies than you need, and that could also be a problem. That could be even as big or if not greater a problem depending on the gene. So here you have this region, you see doubled it, right? You can invert it. Instead of having A, B, C, D, E, F, G, you could have A, D, C, B, E, F, G, H. That's an issue because you could, number one, you could impact the gene itself. Highly unlikely, but it's possible. But the other thing is, by inverting it, they're no longer in the proper place, and so that could lead to all kinds of issues. And then we have a translocation. If you move the gene from one chromosome then to another chromosome, again we run into those problems. They're very similar to having two species mate. You're going to have genes in the wrong place, and when you start to do cell division, especially in meiosis. Why meiosis? Because you're creating a whole new human being. They only have those genes. There's only one set of those genes to begin with, and if there is some kind of mutation in it, then that all the cells in that organism are going to have that mutation. So if you delete it, insert it, translocate it, then every cell in that organism is going to have that translocation, and that could be, usually that means miscarriage, but it could mean some serious issues. Of course, then you have the various somies, right? Trisomy, monosomy, and what we mean is that during meiosis, instead of each cell getting 23 copies or 23 pairs of in the end 23 chromosomes, we have in some cases where you have 3 copies of a chromosome, for instance trisomy 21, which leads to Down syndrome. That's because you have too many copies of the genes on chromosome 21, and that causes cognitive issues as well as some of the other characteristics of children with Down syndrome.